My name is Leticia.
I'm Rodney.
I'm Leanne.
I'm Ryan. And I'm Chris.
And we're Team PANACEA, which stands for Promoting a Novel Approach to Cellular Gene Expression Alteration. So the problem we are aiming to address with our research is that current methods of altering gene expression in mammalian cells is flawed. One of these flaws is that current methods are too harsh on primary cells, which are cells taken directly from living organisms. Electroporation is an example of an existing method, and it involves zapping a hole in the cell so that RNA can enter it. This hole needs to be large enough so that the RNA can enter the cell, but also small enough so that the cell can repair itself and close the hole. However, the hole created by electroporation is most often too large for the cell to be able to repair itself, causing it to blow up. Since these methods are too harsh on primary cells, researchers instead use transformed cells, which are cancer cells that are robust and grow easily. While this is practical for research, it does not show how the treatment will affect primary cells. Another flaw is that current methods are inefficient. Since they are too harsh on cells, they require too many cells and too much material in order to successfully work. Finally, a third flaw is that current methods are non-specific, meaning that cells other than the ones targeted for alteration are also affected by the treatment. An example of this is chemotherapy, which not only targets cancer cells, but also all fast-growing cells, such as hair cells. Thus, in targeting non-healthy cells, other healthy cells that do not need to be altered are also affected unnecessarily. So the goal of our research is to make it easier to alter gene expression to facilitate medical research. And our research question is, how can therapeutic small RNAs be efficiently and specifically delivered into primary cells? The process we will be using to alter gene expression using RNA is called RNA interference, or RNAi. The central dogma of biology states that genes found on DNA are transcribed into mRNA, which are then translated into protein. RNA interference disrupts this process through the use of small RNAs, which is small interfering RNAs, or siRNAs. These siRNAs bind to a complementary mRNA sequence and target it for degradation. Since the mRNA is destroyed, there will be no protein synthesized. Now, Rajan will be explaining how we can get these siRNA to enter a cell of interest.
So the process that we're going to be utilizing to insert the small interfering RNAs into the cell is called receptor-mediated endocytosis, which is a natural cellular process. In this process, cells express receptors on their surface, which bind to ligands, and this initiates a cellular process that results in the formation of a coded pit. Receptors and ligands act as a lock and key, where the receptor is like the lock and the ligand is like a key, and only one lock can fit one key. Once they bind together, it results in the formation of a coded pit on the surface of the cell. Once the coded pit is formed, it eventually is internalized as a vesicle, and once inside, the contents can enter the cell or be degraded or run some other process. We hope to use this to insert the small interfering RNAs into the cell itself. Now we'll take a look at our delivery vehicle protein. It is composed of 3 separate protein subdomains, which is the ligand, the linker protein, and the hepatitis B virus RNA binding protein. The ligand itself is what will confer the cell specificity to our delivery vehicle protein and allow it to enter the cell. The linker protein just binds the other 2 subdomains together, while the hepatitis B virus RNA binding protein is positively charged and will thus bind the negatively charged siRNA through electrostatic interactions. We chose to produce 2 delivery vehicle proteins for our project. The first uses the IL-8 ligand, which is secreted by cells into the extracellular space to attract nearby immune cells. It only attracts a specific subset of immune cells, though, those that express the IL-8 receptor. As a result, using this delivery vehicle protein, we should be able to alter gene expression in only a small subset of immune cells. The second ligand we chose to look at was glycoprotein-1, which is expressed by the marsupial-virus. This glycoprotein-1 ligand binds to the transferrin receptor, which is found in all cells. This receptor allows the import of iron to the cell, and as a result, it's very ubiquitous. And using this delivery vehicle protein, we should be able to alter gene expression in a wide variety of cells. While this does not confer the same level of specificity as the IL-8 ligand does, since we can transfer a wide variety of cells, it could be a useful laboratory reagent for altering gene expression. So the process by which we hope our delivery vehicle will actually facilitate the RNA entering the cell is it will bind to the cell surface receptor, which will initiate receptor-mediated endocytosis and the internalization of our delivery vehicle protein. Once inside, the small interfering RNA will dissociate from the binding domain and enter the cell proper. Once inside, it will bind to the already existing RNA machinery and target its target mRNA for degradation, and this should alter gene expression. So in order to facilitate research, we split it into three main phases. Phase 1 was creation of the expression system, which is an E. coli clone. Phase 2 was producing the deliverable protein itself, and phase 3 was purification of the protein and testing it for its properties and its ability to alter gene expression. We wanted to create an expression system as an E. coli clone because we needed to create a plasmid that would express our recombinant protein. We could not simply buy the plasmid because it was too expensive and uneconomical for the amounts that we need to use for phase 2. So instead, we turned to cloning, which was cheaper and why not easier to do, it was doable for our project. So we started with an already available PIC9 plasmid that was available in the lab, which contained 3 important parts. It has a methanol-inducible promoter, which allows us to decide when we want the cell to express the recombinant protein, because the cell only creates the protein when it is grown in the presence of methanol. The secretion signal, which is the alpha factor, just marks our recombinant protein for expression into the extracellular matrix outside of the cell, which makes it easier for us to collect when we're growing it. And the his4 cycle marker is a genetic sequence that codes for the amino acid histidine, which will allow us to grow these cells in histidine-free media and select for ones that have actually taken out this expression vector as the only ones that can produce histidine. So, starting with this plasmid, we then purchased oligonucleotides from our biotech company that expressed our recombinant protein DNA sequence. We then digested the PIC9 plasmid and the oligonucleotides with 2 restriction enzymes. And here you can see on the left the TARC plasmid, which is PIC9 plasmid, before it's digested, on the right, after it's digested. At the bottom, you can see bands, which marks the inserts, which marks the region that was cut out from the TARC9 plasmid. And it's where, after ligating the TARC plasmid and the oligonucleotide sequence together, we would have a new plasmid that contained the original PIC9 plasmid with the insert, which was our recombinant protein sequence. Once we had this new plasmid, we then transformed E. coli with it to produce our expression vector system, which contained the plasmid that would code for our deliverable protein. And this was the expression system, and the insert was confirmed via genetic sequencing. Now, Key will explain the phase 2 production of the protein itself.
During phase 2, we used the DNA vector from phase 1 to create the delivery protein. We used PKGS115, which is a strain of yeast with a defect in yeast version, which encodes for histidine. Histidine is an essential amino acid, and without it, yeast cannot survive. In our design, his version functions as the selectable marker, making sure only the transformed PKA will grow. First, we grew PKA in media with histidine. Then, through homologous recombination, the correct his version was added to the PKA. Because transformation occurs only by chance, we now put the PKA in media without histidine to only grow those who transform. As you can see in the picture, on the negative control plate, the PKA without our DNA vector did not grow. On the other plate, however, PKA with our DNA vector formed colonies. In phase 1, we also put the methanol in the silver promoter and secreted signal to our DNA vector so that we could control the production of protein. We will pick the colony from the plate and upscale them in the rich media. The expression of the recombinant protein will be induced by adding methanol to the media. Then, because of the secretion signal, the protein will be secreted outside into the growth media. This is the current step we are working on. During phase 3, we will purify and analyze the proteins. We will put the growth media containing our protein of interest through the anion exchange column. And our protein will bind to the column because the RNA binding domain is positively charged, while the column is negatively charged. Then we will elute the proteins using high salt solution. Then we will concentrate the proteins using size filtration, followed by ELISA and SDS phage analysis. Next, we will characterize the siRNA binding using in vitro binding assay and introduce the completed vehicle into actual cells. Now we will test the proteins to see if we can do what it is engineered to do. First, we will measure the level of mRNA using qRT-PCR. Because if siRNA was delivered successfully, then it will destroy the target mRNA. We will then use immunoglobulin assay to label the proteins the mRNA in code form. We hope to see decrease in both mRNA and protein production because it will validate that our targeted delivery system is complete.
By the end of our research, we aim to have expanded on a novel system of delivering therapeutic siRNA into cells in a way that is more efficient, more gentle, and more specific than current methods. The system is also modular, meaning that given the right combination of ligand and siRNA, you can theoretically turn off any type of gene in any type of cell. Looking at our budget, you can see that the cost of our research is quite expensive. This highlights the importance of applying for grants very early in the process so that you can ensure that you have enough money to fund your project. Looking at our timeline, by the end of this fall semester, we aim to finish phase 2 of our research, which is the protein production. Next semester, we will be completing phase 3, which is purification and testing, and in the summer, we'll be analyzing our results from that step. In the fall, we'll be writing and completing our thesis, and finally presenting it in spring 2016 at the thesis conference. Throughout the next year and a half, we will also be continuously applying for grants, expanding on our literature review, and attending conferences. So some of the team challenges we have faced have been found in both our lab work and in our team dynamics. Most of the obstacles we faced were in the cloning portion of the procedure. The size of the plasmid was large, so several adjustments had to be made so that the cloning would be successful. And also, the enzymes were inefficient and took much longer than we had anticipated for it to complete the restriction digestion. In terms of team dynamics, scheduling team meetings has been a challenge because we all have busy schedules and it's hard to coordinate our schedules. But it's important to try to find the meeting times earlier on before the next semester starts so you're not trying to do that last minute. Another anticipated concern we have is writing the thesis, but we aim to resolve this by writing as we go. So once each sub-team finishes their portion of the project, they'll be writing up the methods they used and any results they obtained. And so once each sub-team has completed their work, we will have had most of the thesis written, and we can focus our final year on just editing it. Some advice we have for future teams is to start early, because you never know what obstacles you're going to face or if you're going to have to start a portion of your project over again. So it's important to have that time to account for those situations. Communication is really important so that all members of the team know what's going on and also understand the project, since there are people from different majors all working on the same project. Sub-teams are really efficient for splitting up the lab work and also making sure not too many people are in the lab at the same time. Accountability is important to make sure everybody is fulfilling their responsibilities and contributing to the group work equally. It's important to consult with your mentor. They're a really great resource and they're there for you to give you advice and guidance if you're stuck on one portion of the project. And finally, practice presentations well in advance because it makes you seem more professional and polished, especially if you plan on attending conferences. We'd like to thank our mentor, Dr. Didman, for his guidance and support. We'd also like to thank Dr. Cole, Dr. Skendel, and the rest of the Gemstone staff. And finally, we would like to thank our librarian, Ms. Chankolova. At this time, we will be taking any questions you may have, and we thank you all for listening.
Is there any concern that your protein will be recognized as foreign in relation to, I guess, something in the ecosystem? Like, would there be any kind of immune response to the introduction of your protein, specifically because you're using the hepatitis B or RNA receptor?
At this time, we're just focusing on doing it with primary cells and cell culture. We'd love to be able to see it used in immune systems in the future, but that's a very long way from now.
So you mentioned you're going to use endocytosis to enter the cell, but that is going to confine your construct within another membrane, which is the endosomal membrane. Do you contemplate any alternatives or routes to allow exit into the cytosol where basically the target is going to be located?
I think our previous study has already shown that once it gets in that vesicle, the RNA will leave and enter the cell, and that's already been shown. But the process is not known, but we know that it does happen.
So just looking at the big picture, why would you want to alter gene expression in a mammalian cell? Like, what's the purpose of that?
So if you have a gene, for example, that is related to a certain disorder, you'd want to turn off the expression of that gene so it won't have its detrimental effects.
I noticed that when you talked about the amount of money that your project would cost, I was just wondering like how you calculated.
So we applied to several grants. I don't.
There was the ACC, IAC Fellows Program, and also we
We get some money from Gemstone also.
So because your construct, which is actually quite interesting, has different modular portions which are aimed to exert different functions, if you get a negative result and you don't get to measure, a cell transfection with an effect, are you able to test which of the modules is not working properly? So the binding portion to the siRNA or the portion that binds to the receptor, are you able to distinguish which module is not working?
I believe for the Machikovac, the GP1 protein, we had two constructs planned. So one of them was just like the small portion that could just bind to the receptor, and the other one was the entire protein itself. So if one of them doesn't work, then we do have that backup to see if the ligand